Biology 107 Lab 3, Membrane Structure and Function. In today's lab, we're going to be completing 4 exercises. First, we're going to be making a standard curve of a pigment named beta-cyanin. Secondly, we're going to prepare some beetroot discs. And then we're going to do some temperature stress tests on the beetroot membranes and then some solvent stress tests on the beetroot membranes. So this is a beet. It's a type of vegetable. Maybe you like it, maybe you don't, but it is very useful for studying plant cell membranes. The reason is that the beetroot cells have a pigment in them. It's a purplish, reddish, pinkish pigment called beta-cyanin. It's found inside the vacuole, and the vacuole itself is surrounded in a membrane. That membrane is called a tonoplast. The cell itself is also surrounded in its own plasma membrane. And so if the membranes are damaged, the pigment leaks out. And since the pigment is colored, we can measure how much leakage and therefore infer when membranes are being damaged. This is the structure of beta-cyanin. As I mentioned, it is a purplish, kind of pinkish color. And if you're interested in the chemistry of that, you can see there are lots of double bonds there. And when you see lots of conjugated double bonds in particular, that means you have usually a colored substance that can interact with UV or visible light. So before we get to the part where we use the beets, I want to talk a little bit about a technique called spectroscopy. And so what I have here is a dilution series of the pigment beta-cyanin in 7 different test tubes. And you can see test tube 1 actually has 0, it's just water, and the concentration is increasing. So as the concentration increases, you can see the color gets darker. Now you can sort of eyeball this, and it gives you an idea of what concentration is there. But if I were to look just using my eyes and try to decide what the difference between, let's say, number 4 and 5 is, I wouldn't be able to tell you very quantitatively. So spectroscopy is a method for measuring this quantitatively. In order to understand spectroscopy, it's important to understand a little bit about how the instrument works. So the instrument has a light source, and of course you're going to have your sample solution. You can see it's in one of those little square holders called a cuvette. And the first thing that needs to be done with the light source is it needs to be filtered so that you're only using a particular wavelength of light. So you can see how this works. There's a little lens, it's called a collimator, and that focuses the light on a prism or a grating. And the prism separates the light into basically the different wavelengths, which looks like a little bit like a rainbow. Then what you're going to have is some sort of selector, which is basically a little slit, and it's going to select a particular wavelength of light. So for beta-cyanin, we're going to actually use 525 nanometers of light. That light is going to go through the solution, and on the other side there is a detector. So if the solution is dark and colored, it's going to absorb more light, and this can be measured quantitatively by the actual instrument. I have a little video I'm going to show you that talks a little bit more detail in terms of how the instrument works. I'll play that for you right now.
Here's how a spectrophotometer works. A lamp provides the source of light. The beam of light strikes the diffraction grating, which works like a prism and separates the light into its component wavelengths. The grating is rotated so that only a specific wavelength of light reaches the exit slit. Then the light interacts with the sample. From this point, the detector measures the transmittance and absorbance of the sample. Transmittance refers to the amount of light that passes completely through the sample and strikes the detector. Absorbance is a measurement of light that is absorbed by the sample. The detector senses the light being transmitted through the sample and converts this information into a digital display.
We're now moving on to part A of the experiment, where we're going to make something called a standard curve of absorbance for our pigment. So all pigments are a little bit different, right? They're going to absorb at different wavelengths, they're going to absorb different amounts. So what we need to do is characterize exactly how this pigment absorbs light so that we can make it useful for the later parts of the experiment. So at the moment, what I want to do is draw your attention to table 3-1. And what we're going to do is we're going to make this dilution series. So I showed you those test tubes before, number 1 all the way through 7, where number 1 has just water, number 7 is the most concentrated. And so if you take a look at columns 2 and 3, we're going to measure different amounts of the pigment, beta-cyanin, and different amounts of the water in order to get our dilution series. So I have a video for you right now that I filmed in the lab, and I'm going to play that right now. The first thing that we need to do for this lab is to make a dilution series of beta-cyanine. So I have, I think, everything I need to do this. I have some beta-cyanine, I have some distilled water, I have a micropipetter, I'll get to that in a minute, I have some test tubes, and I have table 3-1. Table 3-1 shows me the volumes that I need of water and beta-cyanine for these 7 test tubes. So you can see test tube number 1, I need 5 milliliters of water, so let's do that right now. In order to measure 5 milliliters, I'm going to use this device here called a macropipettor. I'll bring it closer to the camera, and maybe you can see there's a scale on here. What I can do is adjust the amount of liquid that is measured by that scale by turning the knob at the top. So maybe you can see that yellow line is moving up and down, and what I'm going to do is actually move it to 5 milliliters because I want to measure 5 milliliters of water. The next step I need to do is put this tip on. This tip is where the liquid is going to go into. And then what I need to do is basically depress the plunger at the top to expel the air. Then I can put the tip into the liquid to be measured. Slowly allow the plunger up, and I should have 5 milliliters of water in there. I'm going to deliver it to the test tube by basically pushing down on the plunger, and now I have 5 milliliters of water into test tube number 1. Now what I'm going to do is prepare the rest of the test tubes in the dilution series outlined in table 3-1. So test tube number 2 requires 4 milliliters of water, so I'm adjusting the pipetter. I'm going to pipette the 4 milliliters of water and deliver that to test tube number 2. Test tube number 2 also has 1 milliliter of the beta-cyanine pigment, so I'm going to adjust my pipetter. I'm going to use a new pipetter tip because I do not want to contaminate my different solutions. And I'm going to pipette that 1 milliliter of beta-cyanine into test tube number 2. So there's test tube number 2. You can see it has a little bit of a pinkish color. What I'm going to do now is complete all the other test tubes off camera. Here is our completed dilution series. You can see test tubes number 1 all the way through test tube number 7. You can also see that as the concentration increases, we're seeing a darker pink color going from test tube number 1 to test tube number 7. Now that we've made our dilution series, we're ready to use the spectrophotometer. This here is the Spectronic 200. So this is the instrument that we have in the Biology 107 lab. There are many different models of spectrophotometers. These ones are actually really nice to use. And if you take a look at that, there's a few different things to point out. First of all, there's a wavelength control, so I can control which wavelength of light is going to be measured in the instrument. So we're using 525 nanometers. There's a digital readout, and there's a sample compartment. I'm going to show you how to use the spectrophotometer in a video right now. Now what we are going to do is use the Spectronic 200 to take the absorbance of our samples. So there are our samples. What I've done is taken them and I've put them all into these cuvettes here. These cuvettes are kind of like little square plastic test tubes. You can see this here is number 1, and it's ready to go, and that's actually the one that we need. So the cuvette is going to go inside the Spectronic 200. It's going to go here into this holding compartment. And what we're looking at here is on the left is a light source, and on the right over there inside that little hole is a detector that's going to read the amount of light that's going to go through that sample. I'm going to shut the lid and just make sure everything is ready to go. You can see it's set at 525 nanometers. I can change my wavelength if I want to by turning this knob, but we want it at 525. And since this is my water, this is my reference or my control sample, so what I need to do is calibrate the device by pressing this button here. So the 0 button, what it does is basically tells this device what the baseline is. So since water is our control and there's no pigment in there, the water sample should read as 0. So that's what I just did to the device. I told it to read the water sample as 0. So you can see there is my first reading. So now what I've done here, this is test tube number 1. I can write this down in my table 3-1, and there you see I've written down 0.00 for test tube number 1. So now I can read number 2.
So I will take out my sample. I will grab cuvette number 2. You can see there's number 2. I'm going to put it in the device. I don't need to calibrate it anymore. I just need to shut the lid and then take the reading. So you can see what my reading is, 0.26. I'm going to write that down in my table, so 0.26, and proceed to take the absorbance of the rest of my samples in the same way.
Now back to table 3-1, and we have a little bit of data now. So you can see I have all my absorbances there in the final column. You will have to determine the concentrations and the dilutions. That is going to be your job. If you take a look in the lab manual, there are instructions on how to do so if you're not sure how to do these kind of calculations. You can always contact me if you have any questions. So what we're going to do with this data is we're going to make a plot which is called a standard curve. So to make a standard curve, what we need to do is plot absorbance and concentration on a graph. By the way, you can make your graph on graph paper, or you can use a computer software package. If you do choose to use software and you're interested in using Microsoft Excel, I'm going to have a small Excel tutorial at the end of this video that will hopefully help you to get started. So take a look at that, and you can always contact me if you have any questions about Microsoft Excel. So we're going to plot absorbance on the y-axis. So there's absorbance. Absorbance does not have actual any units, but you do need to indicate what wavelength the absorbance was measured at. So that would be 525 nanometers. Concentration is on the x-axis, and the units are micromoles per liter. Notice that's a big M for moles per liter. So plot the data. There is the data right there, and make sure you use big visible symbols. Don't use little dots or something that are hard to see. I want to see all the data points nice, clear, and visible. Then what you're going to do is add a trend line. So a trend line is not connecting the dots. A trend line is a line of best fit, so the whole thing is straight. You can use a ruler on graph paper, or there are ways to do this on Excel. And the nice thing about Excel is Excel will actually give you the equation of the line. So y equals mx plus b. There is no b because the intercept is 0, 0. So y equals mx, therefore the slope in this case is 0.05x. So take note that this plot can be used later, and that's why we're generating this plot. We're going to use it later in part C and D, and we're going to use that to determine our concentrations. So in part C and D, we're damaging the membranes using temperature and solvents, and we're going to measure an absorbance. And from that absorbance, because we don't know the concentration, we're going to determine the concentration. I'll show you basically how this works. Now, the relationship between absorbance and concentration is called Beer's law, or the Beer-Lambert law. This is named after a couple of scientists who initially investigated this. So if you take a look, there's a formula. Absorbance is equal to epsilon B C. So C is concentration. So what is B? B is actually the width of the cuvette. The cuvette is the little square test tube-like thing that the sample is in, and those are standardized to be 1 centimeter. And epsilon is a constant. So what it's saying is that A is directly proportional to C, the concentration, under given conditions for a given pigment. So more on this later after we complete parts B, C, and D. We're going to come back to this graph and show you how to determine concentration when you have an absorbance value. So part B is preparing our beetroot discs. So we have a beetroot here, and what I'm going to do is basically cut it up and make these little puck-like discs. I have a video for you that I'm going to show you right now of me preparing these. So what we're going to do now is prepare our beetroot discs. So I'm going to use this little device here. This is called a corer, meaning it makes cores. So basically what I need to do is stick it into the beet, and something like this. It's going to grab basically a chunk of the beet, and I can push it out using this little device. And there we go. Now I have a beetroot core. So I'm going to cut those up into small sections about 5 millimeters in length. So there are my beetroot discs. The last thing I need to do, you can see I'm getting a lot of the pigment all over my fingers.
and there's going to be a little bit of leakage of this pigment. So what I'm going to do is wash them. So I have this little bag that we've made just out of screen. These beetroot discs are going to go into the bag, and what I'm going to do is rinse them in water for about 10 minutes. You can see already some of the pigment is leaking out, and after the 10 minutes this should be quite clean. So here they are, the beetroot discs are now finished being washed, and now they're ready for the temperature and solvent experiments.
Now that we have our beetroot discs, we're going to use those discs and we're going to subject them to different conditions to see if we can damage membranes. So here's how that's going to work. There's my little discs. I'm going to be sticking them in different temperatures in water baths for 2 minutes. So you can see that water bath there is 65 degrees Celsius. Afterwards, the disc is going to go into a test tube of water, and it's going to be mixed. I'm going to mix it at 5, 10, and 15 minutes. And over time, if the membranes are damaged, they're going to leak some pigment, and the water is going to change color. Like I said before, I can't really tell how much pigment is in the water using my eyes, so that water is going to be transferred to a cuvette, and the cuvette is going to be measured in the spectrophotometer, so I can get an absorbance, get an exact value of how much leakage I've had from that particular sample. I have a video that's going to show you me doing the temperature stress test, and I will play that for you right now.
So we're now ready for the temperature stress experiments. So you can see what I have here is I have a Ziploc bag. It's labeled with the temperature that I'm going to do, 65 degrees Celsius. And inside the bag right here, I have one of the beetroot discs. So what I'm going to do is stick that into this water bath here. This water bath has been programmed at 65 degrees Celsius. It's not quite 65 degrees. Sometimes when the lid is off, it has a hard time maintaining the proper temperature, but it's close enough for the purpose of this experiment. So what I'm going to do is take the plastic bag with the beetroot disc, it's all sealed up nicely, and I'm going to subject that beetroot disc at 65 degrees for 2 minutes. So the 2 minutes are up. I'm going to take the beetroot out of the water bath, and what I'm going to do is put it into this test tube of water. So here's my beetroot disc. You can see it's in a test tube of water, and what I'm going to do is give it 15 minutes for any of the beta-cyanin that's been ruptured from the beetroot cells to get extracted from the beetroot cells into the water. And to help this out, I'm going to use this instrument here called a vortex. So what I'm going to do is vortex, which is basically stirs it up a little bit every 5 minutes over the 15-minute period. So here's how the vortex works. I put the test tube in the vortex, I press it, you can see it stirs it a little bit. Vortex at 5 minutes, and vortex at 10 minutes. So here we are at the 15-minute mark for our final vortex. Mix that up a little bit. And if you take a look at our solution with this white background, you can see that the solution has become a little bit more pink, which means some beta-cyanin has been released over the last 15 minutes. So I'm going to take a break now, and I'm going to do all the other samples, and then we'll take a look at the whole batch of them together. So there's the rest of the experiment completed. You can see I have all the temperatures done. They're all conducted the same way the 65 degrees Celsius was done, so they were subjected to each particular temperature for 2 minutes, and then allowed to extract the beta-cyanin for 15 minutes with vortexing in water for each of these test tubes. So there's our solutions from the temperature stress test, and I've poured them into the cuvette, so you can see there's definitely a little bit of a pattern there. So let's take our readings in the spectrophotometer, and then we'll have a chance to analyze the data. The first thing we want to do is add our blank solution, so that's the one that's just water, into the instrument, and make sure that it does read 0, and it does not. So I want to make sure that I do 0 the instrument so that it's ready to read my samples. So now that the instrument is calibrated, I can open this up and I can take the readings of all of my samples, just like what I did before. So I remove the blank, I'm going to put in the sample for the -20, shut the lid, and you can see there's my reading for the -20. I will complete all the readings for the other samples.
Part D is the solvent stress tests. The solvent tests are a lot like the temperature tests in that we're going to take some beetroot discs and we're going to subject them to some different conditions. In this case, what I'm going to do is subject my beetroot discs to different solvents, so water, methanol, ethanol, and propanol. So you're probably wondering what is the difference between these different solvents. You can see from the chemical structures that the difference from each is basically a trend of one carbon atom. So water has no carbon atoms, methanol has 1, ethanol has 2, and propanol has 3 carbons. So what does this do to the chemistry? This is going to be one of the questions for your report, and I'm going to give you a little bit of the answer right now, is that basically this is a trend of polarity. So water is the most polar out of these molecules, and as we move towards more carbons, we're making the whole molecule a lot more hydrophobic or less polar. So we're looking at the effects of the polarity of the solvent on the structure of the membranes. Once we have the discs in the solvent, we're going to leave them there for 30 minutes. So just like before, we're going to mix them a little bit as we go along. So every 10 minutes, I'm going to vortex these test tubes. At the end of that, just like what we did with the temperature stress tests, I'm going to take the absorbance of the solution to see how much of the pigment has leaked. I have a video for you right now of the solvent stress tests.
So if you take a look, what I have there are 4 test tubes, and each of them has a solvent in them. I have 5 milliliters of water in the first one, 5 milliliters of methanol, 5 milliliters of ethanol, and 5 milliliters of propanol. So for the solvent stress test, what I'm going to do is I'm going to add a beetroot disc to each of the solvents. And this is going to sit for a total time of 30 minutes. So a little bit longer than the temperature stress test. Basically what they're going to do is I'm going to vortex them just like before for at 0, 10 minutes, 20 minutes, and 30 minutes. And at the end of that, we will take the absorbance just like we did with the temperature stress tests.
Here are the results from our solvent stress test. You can see I have the test tubes there on the left and the cuvettes on the right. So let's take a look at our data and see what we're going to do to analyze this data. So here's our data from the temperature tests and the solvent tests. So you may notice that we still have not figured out what the concentration is of our beta sign and leaked in each of these different conditions. So remember, I told you earlier that what we need to do is figure out these concentrations using our standard curves. So I'm going to show you how to do that right now. So in order to determine our concentration using the standard curve, we can either plot the data on the curve or we can use the equation of the line. I will show you how to do both methods right now. So here's an example. We have a sample and the absorbance is 0.25. So what you can do is basically plot that on the graph. So 0.25 is right about here. And you can see I've plotted it over to my trend line. And then basically, in order to find out the concentration, I can draw a vertical line down to the concentration. So in this example here, the concentration is about 5 micromoles per liter. So that's one way to do it. This is maybe a little less accurate than using the equation of the line, and I will show you how to do that right now. So to use the equation of the line, I'm just using that y equals 0.05x. So in this particular example, absorbance, of course, is y, and x is concentration. So I can manipulate that formula and turn it around so that concentration is absorbance divided by 0.05. So in this case, that's 0.25 divided by 0.05, and therefore my concentration is 5 micromoles per liter. So you can use either method. Like I said, if you have the equation on the line, it's pretty easy to punch that in. And do make sure you do provide me with a sample calculation for your lab report. So here we are back to our tables. I'm not going to give you the values for concentrations. That's something that you are going to have to do yourself. But once you get your concentrations, you're going to produce some graphs for me. So I'll show you how to make those graphs. For your temperature stress test, you are going to make a graph, and it is going to have temperature on the x-axis, concentration on the y-axis.
So let's add some data points. I'll make a little graph here for you. So minus 20, some of the other temperatures are going to all have data points. And so for this graph, what you're going to do is basically connect the dots. You can do a straight line or you can do a curved line like what I just did there. There are different ways to do it on the computer, but what you're going to see is a trend. Now, this is not the trend that you're going to see. You're going to see a different trend. I will leave that as a surprise for you. But what I do want to do is give you some graphing tips. First graphing tip, use clearly defined data points. So you can see what I did is I had some nice circles as my data points. They don't have to be shaded in. You can have them open. You can use squares, you can use triangles, stars, and so on. Just make sure your data points are really clearly defined, that you're not using the little teeny tiny ones that are hard to see. I want to be able to see every data point very clearly. Number 2, include the proper units. So you can see that I have degrees Celsius for temperature and micromoles per liter for concentration. Make sure you have the accurate units or else temperature is meaningless. This could be Fahrenheit or Kelvin or anything like that. Number 3, put a title below the graph. So in Biology 107, a graph or a figure are the same thing. They're all called figures, and figures have the titles below. A table is a different thing. A table has a figure above it. So a figure title is below, and that figure title is going to have a number. So that's the next point. And so that number is going to be, if you have 3 graphs, they're going to be 1, 2, 3. Or in this case, it's lab 3, so I've named them figure 3-1, 3-2, 3-3. So make sure you have a system and they're numbered and it makes sense. Secondly, make sure that the title is descriptive. So this particular experiment is involving membranes and beats and temperature and beta-cyanin. So all those words should be in your title. Give me a description of what the experiment was about. If you can't do it in 1 sentence, do it in 2 sentences. It's not really a big deal. Do make sure that you include the species that we were researching. So in this case here, it was a beetroot, but you want to use the scientific binomial name, and that of course is going to be in italics. For the solvent test, you're going to make another graph, and this graph is going to have solvent on the x-axis and concentration on the y-axis. So this graph is going to be a bar graph. So I'll make some bars here. Here's the water. Going to shade that in to make it a little bit easier to see the bar. Here's the methanol. And you're going to make yours look a lot prettier than this. Maybe use a ruler if you're doing this by hand, because we want to have a nice and tidy graphs. So same thing here. You're going to see a trend that you're going to be able to describe. This is not the trend that you are going to see. But you're gonna make a nice graph, and you're gonna see a nice trend, and you're gonna be able to discuss that in your lab report. So for your lab report, you're gonna have 3 graphs. The first one is gonna be the standard curve. The second one is gonna be the temperature test. The third one is gonna be a solvent test. All of them are gonna be beautiful, and you're gonna get lots of marks in your lab report.
Let's talk about this Lab 3 report. This Lab 3 report is going to be a formal lab report. So this is a pretty major assignment. It's in fact worth somewhere around 14% of your final grade, and it is going to take quite a bit of time, particularly if you've never done a formal lab report before. This is a pretty major assignment. So this formal report is going to include the following sections: a title, introduction, methods, results, discussion, literature cited, and appendix. And to give you an idea of how long this is going to be, you can see there's an estimation of how big those sections are going to be. So it's going to be about 5 to 6 pages of actual written material, double-spaced, typed, and probably about another 6 pages of all of your other stuff. So remember, you're going to have 3 graphs, that's 3 pages. You're going to have a literature cited, you're going to have some rough data and calculations, and so in the end, most people hand in something that's somewhere around 10 to 12 pages of work. Now, I'm not going to talk about in this video how to do all these sections. That would be a whole nother probably 40-minute video, but what I am going to do in the next few lectures over the next 2 weeks is talk about each of these sections one by one at the beginning of class. So it's important that you come to class on time and listen to all of the lab report tips that I'm going to give to you. Here are some tips to get you started. First, read the report instructions. I'm going to give you some instructions with today's lab, plus if you look at Appendix A, There's lots of detailed instructions on how to write and do these lab reports. Number 2, always be specific. So what I want you to do is get practice in this class giving me good, specific, clear, scientific, technical language. So you can see in this case there's a couple of examples there. You can see 2 sentences. The highest temperature caused the most damage. The second one says, the highest temperature, 85 degrees Celsius, caused the most damage with 12.4 micromolars of beta-cyanide. So which one's better? Clearly the second one is better. It's giving me details. It's not giving me vague answers. It's telling me exactly what temperature, exactly how much damage, and then furthermore it's referring me to figure 2. So if I want to see more specifics, I can go to that exact data. One more quick tip is learn how to use special characters in your word processor. Okay, if you're going to talk about carbon dioxide, it's got to have a little 2. If you're going to talk about micromolars, it needs to have that special symbol, and so on. This is just something that makes your report look a lot more professional and clear and precise. So a couple more tips for you. First of all, it is very important to know that you will not be able to do this report in one day. Maybe you could, it's just not going to be very good. What I suggest is that you work on this over several days. So maybe one day you work on the introduction, another day you work on the results, another day on the methods, discussion, and so on. Then you have lots of time to proofread, put things together, and consult with me. So I make this offer to all my students. If you have any rough drafts that you want to show me, either the complete report or just different parts of it, that you do come to me and show me what you have. I'd be happy to help you with any sections, whether it's just understanding how to make graphs, interpret the data, or with your writing, or anything like that. Just contact me during my office hours, and I'd be happy to do so. So this is the end of Lab 3. I do have a Microsoft Excel tutorial after this, but if you're comfortable using Microsoft Excel, you certainly don't need to watch that. If you're interested in seeing how to make the graphs using Microsoft Excel, then just keep playing this video and I'll play that for you right now. So let's talk about how we can make graphs in Microsoft Excel. So here's Microsoft Excel. You can see I have some data there, some concentration and absorbance data. This is not our data. This is from another experiment, but we can do the same thing to produce a graph. So what you need to do is basically select the data. And so what you can do is bring your mouse over here and highlight all of that data in both columns, concentration and absorbance. Then you're going to go over here to insert, the insert ribbon. You're going to go over and you can see there's these different graph types over here. You're going to select scatter graph, and this is the one I like the best. And this way it doesn't draw the lines because I'm going to draw the trend line afterwards. So I'm going to select that. So first we need to just fix this up a little bit. Notice there's a title up at the top. We don't want titles at the top in Biology 107, so I'm just going to click on this and then hit delete to remove that. So you can put your title down at the bottom if you like. So figure 3-1, give it a descriptive title. Or you can put the title in Microsoft Word after you're done inserting the graph as well. So notice it does not have any labels on the axes. So what you can do is click on the graph itself. And if you take a look, now this has made an extra section up at top. So we have chart design and format. I want chart design. And over here on the left, you can see add chart element. So over here, axes titles. And I'm going to add a horizontal axis title. So there it is. So remember, my horizontal title is concentration. And my units are micromoles per liter. And so to get that micro, I'm going to go up here to insert. And somewhere there should be a symbol. It's over here on the right. And you can see symbol. So what I need to do now is find the micro in here. So I'm sure it's in here somewhere. I just have to see if I can find it. Scroll down a little bit. It's probably here somewhere. Where is it? Oh, it looks like it's one of my recently used symbols, so I'll just grab it there. And I hit insert and close, and there is my special symbol. So if I'm not happy with the font size on this thing, I can select the entire graph and I can just increase the font size on everything. That looks a little bit better already. So I need to add the horizontal title, so chart design again, add chart element, axis title, and vertical. So my vertical title is, of course, absorbance. Type that in, and we got 525 nanometers. There we go. So you can adjust these things, move them around a little bit. If you don't like the size of your graph, you can always resize it, and so on. And there we go. So something you can see that automatically gives me some nice symbols. If you don't like these symbols, there are ways to fix them. I think this is under Format. Or if I double click on it and go over to the right, there are some options here for my series symbols and series options. So I can click on this and right here. So the marker and marker options. So in this case here, let's see here. I'm going to maybe make it a different shape. I'll make it a triangle. And let's see here, maybe I want a different color. What color do we like? Maybe I'm out of blue ink, so I will choose, let's say, red ink. And if you don't like the size, you can increase the size. So there we go. Okay, so there's my graph. And I mentioned before that one thing that I like to do is add a trend line. And that way I can get the equation of the line as well. So what I do is I double click on this, or sorry, no, I don't double click. I right click on this. And once I'm on the data, as you can see down here, it says add trend line. So I click on add trend line, and if I scroll down here, you can see I can put display equation on the chart. So there's my equation right here. And so you can see y equals mx plus b is y equals 0.621x minus 0.002. So something you can do for this graph is with that trend line, so let me just go back to the trend line menu here, is I can also set the intercept to 0, 0, because of course the graph does go through 0, 0. So what this does is corrects the line a little bit, gives me slightly different slope, but it removes any issues I have with some really, really small numbers. So I can do that, it removes the intercept, and I just have y equals mx, which is 0.0619x. So that's how to make a graph. If you want to make a bar graph, you could do something similar, except for when you select the graph type, you're going to select bar graph. So play around with it a little bit, and hopefully you can make some nice graphs for this report.